Hello, welcome you all to the family of BioGoogle. In the previous video, we have discussed about the formation of the nucleosides and formation of the nucleotides. Meanwhile, in DNA as well as in RNA. Okay, let us recall the things and we'll move on to the structure of these nucleotides. Means how the pentose sugar, nitrogen base, phosphate, all three combine together to form a nucleotide. Okay, so firstly what happens? Pentose sugar, that is deoxyribose in DNA, ribose in RNA. They combine with adenine to form deoxyadenosine. Means deoxyribose plus adenine, that becomes deoxyadenosine. With guanine, deoxyguanosine. With thymine, deoxythymidine. With with cytosine, deoxycytidine. Okay, then all these are called nucleosides of DNA, also called deoxyribosides. Deoxyribosides or the nucleosides of DNA. And to these nucleosides, when the phosphate group binds, we add the word monophosphate. One phosphate is added. Okay, so deoxyadenosine plus phosphate is called deoxyadenosine monophosphate. Then deoxyguanosine plus phosphate, deoxyguanosine monophosphate. Deoxythymidine plus phosphate, deoxythymidine monophosphate. Deoxycytidine plus phosphate, deoxycytidine monophosphate. So these are the nucleotides of DNA, also called deoxyribotides. Here you see the word deoxyribosides, deoxyribotides, nucleosides, nucleotides. But when we come to RNA, here the sugar is ribose. It was deoxyribose in DNA. In RNA it is just ribose. So ribose with adenine, adenosine, with guanine, guanosine, with uracil, uridine, with cytosine, cytidine. So adenosine with phosphate, adenosine monophosphate. Guanosine with phosphate, guanosine monophosphate. Uridine with phosphate, uridine monophosphate. Cytidine with phosphate, cytidine monophosphate. Cytidine monophosphate. So these are the nucleotides of DNA. These are the nucleosides of RNA. These are the nucleotides of RNA. Okay, tides of RNA. Nucleotides of RNA. These are the nucleosides of DNA, nucleotides of DNA. Okay, and in today's class we will discuss about the structure of this nucleotide. Means how this nucleotide is formed and what are the bonds involved in all these three. Means during the formation of a nucleotide, maybe deoxyribotide or a ribotide, we'll see how the different bonds, maybe a glycosidic bond, maybe the ester bond or a phosphoester bond, or else the hydrogen bond will help us in studying and understanding the formation of the nucleotide. Okay, we'll see the structure better once you pause the video and take down the things. They will help you in examination. So you may have to remember all these three. Okay. One more thing, we have heard about a term called ATP. Till today we just learned that ATP is energy molecule. What is it? What it is? Yes, ATP means adenosine triphosphate. We have ATP here only. Okay, so adenosine plus one phosphate is called adenosine monophosphate. One mono. One mono, 2 biphosphate, 3 3 triphosphate. That's it. Okay, means for this adenosine, if one phosphate binds, it is called adenosine monophosphate. To this adenosine, if 2 phosphates bind, it is called adenosine biphosphate or diphosphate. If for this 3 phosphates bind, it becomes ATP adenosine triphosphate. So the energy molecule, universal energy molecule ATP, it is nothing but the nucleotides of RNA. ATP is a trinucleotide of RNA, remember. Okay, so we will see how different structures are arranged together to form a nucleotide. So first of all, you all should remember till the end of this chapter that the DNA is made up of 3 major structures. DNA or RNA is made of 3 major structures. Okay, so we'll see the nucleotide formation of a nucleotide. Formation of a nucleotide. Right. So whatever we have discussed is pentose sugar with the nitrogen base will form a nucleoside. With the nucleoside, when we bind a phosphate, that is phosphoric acid, that becomes a nucleotide. So pentose sugar plus nitrogen base will form nucleoside. Nucleoside plus phosphate will form nucleotide. So we will see this sequence with structure. Okay. So for that we should draw a pentose sugar. Pentose sugar with 5 carbons. Among 5, 4 carbons are there in the ring. Other one is in the form of a long R. Okay. So OH, H. Remember, it is H. This is also H in case of DNA. This is OH in case of RNA. This is actually CH2H. But when we write towards the left side, it becomes H2COH. Okay, don't confuse it, it's H2COH. It is CH2H only. But we are writing CH2H in this form. Okay. So don't confuse with that. CH2OH, and here we have a H. And let us label the carbon numbers. The first carbon, second carbon, third carbon, fourth carbon, and this is the fifth carbon. Okay, this is our pentose sugar. Right. To this pentose sugar, the nitrogen base should bind. I have told you in previous class, while differentiating the purines and pyrimidines, the purines will form 1' 9 glycosidic bond. The pyrimidines will form 1' 1 glycosidic bond. It means when the nitrogen base binds to the sugar here, the first carbon of the sugar is attached and 9th nitrogen is involved in the formation of the glycosidic bond. First carbon of sugar, 9th nitrogen of the nitrogen base. Similarly with pyrimidines like a guanine and thymine and cytosine, first carbon of the sugar and first nitrogen of the pyrimidine. Okay, so they will bind here. So if I write adenine, it won't be that good, it will form bond. This bond is a glycosidic bond, adenine. Pentose sugar, nitrogen base, but we'll draw a structure of adenine for understanding purpose. Then it's easy for understanding. So here we have So this is our structure of one of the purine. So we have a nitrogen at first position, 1 2 3 4 5 6 7 8 9. So we have a nitrogen here, and for this nitrogen there is always a H. Pentose sugar, nitrogen base. Okay, so here the sugar will donate the OH group, nitrogen base purine will donate the H, and they will release one molecule of water, that is H2O is released. H2O. Okay, so this bond is known as glycosidic bond. This bond is known as glycosidic bond. Okay, so glucose, sugar, nitrogen base, both will form a bond known as a glycosidic bond. The bond is formed between first carbon of the pentose sugar and 9th nitrogen of the purine. So 1' 9 glycosidic bond. So now this structure is called a nucleoside, because there is a fusion of a pentose sugar and a nitrogen base, where they will lose one molecule of water. That is a dehydration. Process is a loss of one molecule of water is called a dehydration, and the bond formed is known as a glycosidic bond. So now the nucleoside is formed, right? And now for this nucleoside, one phosphate will bind. Okay, so the phosphate will bind at the third position or fifth, in both the positions. Okay, one phosphate here, again next sugar, next phosphate will bind. Okay, we'll see how this phosphate will be bound here. Phosphate is H3PO4, and I told you we can write H3PO4 in the form of OH double bond O, H3PO4. We can write H3PO4 in this form, right? So we'll write this form here, H3PO4. So P OH double bond O, OH, phosphate. Okay, here the sugar will donate the hydrogen, phosphate will donate the OH group. Again there is a loss of one molecule of water. And this bond between phosphate and sugar, and this bond is called ester bond or phosphoester bond. It is called ester bond. Okay, now this structure is called a nucleotide, where we have a pentose sugar, we have nitrogen base, and we have a phosphate group. So this structure is now called a nucleotide. Again, look at the diagram properly, look at the structure properly. When the sugar binds to the nitrogen base by losing one molecule of water, it is called a glycosidic bond, and this structure is known as a nucleoside. And for this nucleoside, when phosphate binds, and this structure is now called a nucleotide, and this bond is called ester bond or phosphoester bond. Then the question is, how the next nucleotide will attach here. How the next nucleotide will attach here. Okay. So here we have written CH2H of this sugar. We'll write one more sugar here. We'll write one more sugar here. CH2OH. H2C. So we can write like this. Is it okay? CH2H. C H2 OH. CH2H is there. So now we can write the sugar. We can write the sugar as per the norm. So we'll join here one of the nitrogen. We have written the adenine. One of the purine will take just simply the cytosine. No to make the complex. This is glycosidic bond here, right? And here we have another phosphate like this, joining with another phosphate bond. Now this is another sugar. This is another sugar. Okay, 1 pentose sugar here. So now this OH will, this phosphate will donate OH group. This will donate the hydrogen. And now another phosphoester bond is formed. Another phosphoester bond is formed. Phosphoester bond is formed. Again there is a loss of one molecule of water. Okay, and now look at the diagram. One sugar, between the two sugars there is a phosphate. Between the two sugars there is a phosphate, and on either side of the phosphate there is one ester bond. There is one ester bond. Phosphodiester bond. Phosphodiester bond means two ester bonds are formed on either side of the phosphate. So this complete bond is now called phosphodiester bond. Phosphodiester bond. Okay, phosphodiester bond. Phosphodiester bond. One phosphate, two esters on either side of the phosphate, joining the two sugars or two nucleotides. Joining the two nucleotides, and this structure is now called a dinucleotide because there are two nucleotides joined by the phosphodiester bond. This is one nucleotide with sugar nitrogen base phosphate. This is another nucleotide with sugar nitrogen base phosphate, and they are bound by one of the phosphates. Okay, this is how the nucleotide is formed. This is how the nucleotide is formed, and many nucleotides will join each other. Okay, I am telling you, remember the structures if you can remember, otherwise don't try to remember. Okay, I have another easy method to understand the bonds. That's enough for examination purpose. Okay, even for a NEET exam, that's okay, right? Don't make yourself complicated, because you are going to study again this chapter in second PUC also, means in chemistry also. In a chemistry, that is in a biomolecular chapter is there. In chemistry we understand all this in detail in chemistry structurally. Okay, otherwise just try to understand this method. A very interesting concept it is. Sugar binds with a base by losing one molecule of water by forming a bond, a glycosidic bond. Nucleoside binds with the phosphate by losing a water molecule to form an ester bond, to form a nucleotide. And this nucleotide will bind to another nucleotide by using another phosphoester bond to make phosphodiester bond and to make a dinucleotide. Okay, this is how the one nucleotide, mono, dinucleotide, tri, tetra, penta, like that many nucleotides keep on joining each other, one another. Okay, so for you better I will tell you a simple method. Okay, if you want, pause the video, take down the structure. I will go for a simple method rather than writing all these structural details, then we will understand the process easily. And one thing, one code I'm going to use here. We have 3 bonds here. One is a glycosidic bond. I am just using the word B1 in glycosidic bond. Then a phosphodiester bond. I am using the word B2 in diagram. There is another bond called hydrogen bond. I use the word B3 while drawing the diagrams so that I can reduce the space over here. So I will just write a sugar. And I will take adenine. Next I will take phosphate. Sugar, adenine, nitrogen base, phosphate, one nucleotide. Similarly, another sugar. Next not phosphate, we have a next pentose sugar. Next we'll take a guanine. Next we'll take a phosphate. So one nucleotide, 2 nucleotide. So now this bond is B1, means glycosidic bond. This bond is B2, phosphodiester bond. Then where is B3? Okay, we'll add one more nucleotide right here for understanding purpose. So here we have another nucleotide. So with another phosphate, right? We'll take one more. We'll take cytosine here. This is one strand of DNA. Hydrogen bond is used to join another strand over here. But that strand is reverse. This strand is facing towards upside. The other strand will be facing downward. So I will try to write here. So in front of cytosine it will be G. In front of guanine it will be C. In front of adenine it will be thymine. Okay, so I will just take this way. And here I can take over this. Sorry. You can take like this. And for this so this is how we can draw another strand of the DNA. Another strand here. We'll write phosphates over here. So phosphate will be here. Phosphate will be here, right? sugar, nitrogen base, phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base, phosphate, phosphate. Okay, this is one strand of DNA, this is another strand of DNA. Okay, you should also know that adenine always form 2 hydrogen bonds with a thymine, guanine always forms 3 hydrogen bonds with a cytosine. So we have adenine, thymine, 2 hydrogen bonds, guanine, cytosine, 3 hydrogen bonds, cytosine, guanine, 3 hydrogen bonds. This is the structure of DNA. One strand running in this direction, other strand running in this direction. Okay, so we can now label the ends of the DNA. You can label the ends of the DNA as this as the 3' end, this as the 5' end, this as the 3' end, and this as the 5' end. Okay, this is how the nucleotides join together to form a polynucleotide with 2 chains. One chain, one polynucleotide, 2 chain, 2 polynucleotide. Both together will make a double helix. They will keep on turning, double helix. So we have one nucleotide here, we have a 2 nucleotide here, we have a 3 nucleotide here. Similarly here we have one nucleotide, we have a 2 nucleotide, we have a 3 nucleotide. So 1 base pair, 2 base pair, 3 base pair. It means pair of 2 nucleotides is 1 base pair. Pair of 2 nucleotides, 2 base pairs. Pair of 3 nucleotides, 3 base pairs. Like that, the base pairs will be formed each other or one another, one above the other. Okay, this is how nucleotides, monomers keep on joining each other to form a polynucleotide or a polymer. Right? That's how the structure is formed. Right. In the next class, we'll discuss about the Chargaff's rules. Chargaff's rules means one of the scientists called Erwin Chargaff, he has made some rules for the arrangement of the nitrogen bases. Arrangement of nitrogen bases means the number of nitrogen bases, quantity of nitrogen bases, number of bonds, etc. So there is a rule, set of rules called Chargaff's rules. We will discuss in the next video. Also, we will find some types of DNA. Some types of DNA in further videos. Lastly, we will go to the structure of DNA, that is double helical model of DNA proposed by Watson and Crick. Right. If you again have a doubt in this concept, better you go through the video again or else write the question in the comment section. For any more doubts, I will give my contact number at the end of the chapter. Okay, and if you are new to my channel, please subscribe to my channel. Okay, if you like the video, share the video first of all to your students, if you are a teacher, to your friends if you are a student, or anyone who is in need of this chapter. Okay, maybe the preparation of the CET or the preparation of the NEET examination, and if you are taking the long-term coaching, it will be very much helpful. And if you are that much interested, I can take up the complete syllabus in further days. Okay, and if you want any more specific concept in any chapter, at earlier, of course, as per schedule I will make the video, but if you want that particular video, particular concept very soon, please let me know in the comment section so that I can prepare the video for you only. Okay, till that, keep learning. See you in the next video. Thank you.